Hello, I am Dr. Eugene Aiello. I am a practicing physician. I am also an author and a researcher focusing on biophotonics. Starting this video series because I want to elaborate on concepts that are in my book, The Conscious Canary, a complete model of biophotonic communication and multicellular life. It sounds complicated. At the base, what I want to get through in this series is that much of biology, which is extremely complicated with chemical reactions and different interactions of charged particles, is actually way less complicated. That life is actually based on charge separations. And charge separations are ways that the cell can use electron flow to have controlled reactions. And what I mean by that, from everything from protein changes to different gradients set up within membranes for action potentials and nerves, multiple, even genetic expression is going to be controlled by different ways that electron flow and charge separation dictate physiology. So the first thing I want to talk about is what is charge separation. Life equals charge separation in my mind after my research. Biophotonics plays a huge role in creating initial charge separation and controlled charge separations within the cells. So an example of a charge separation would be when you would add an electron to oxygen. When you do that, you're creating a negative oxygen molecule. So now instead of a neutral oxygen, now we have a negatively charged particle that can donate electrons or move electrons. So you can create a gradient of electrical flow based on having a charge separation. Another example is a charge separation that's held within ATP. ATP we're going to talk about in a minute, but ATP, adenosine triphosphate, is the currency of energy in the cells. This is how our cells store and use energy. So ATP is a stored charge separation that when it's broken down into ADP and P, it's allowing an electron to escape and transfer. Why is this going to be important? You're going to see. We're going to go through biologic models of how things occur for energy production and multiple other models, and it's all based on movements of electrons. Electrons have to be moved in a controlled manner, and if they're not, they could be dangerous. So different mutations, different proteins can be denatured, lipids. So having that electron flow in a very specific way is how life controls this flow and doesn't damage itself. So we're going to start out talking first about single-celled organisms. You've got two different types. You've got photosynthetic and non-photosynthetic organisms. Photosynthetic organisms directly harness electromagnetic energy from the sun to create a charge separation, which is then utilized to create charge flow, which eventually ends up in ATP production. So I'm going to walk through it slowly. So within the chloroplasts of a photosynthetic organism, there's two photosystems. Each photosystem is harnessing this electromagnetic energy for a different type of charge separation. So I'm going to start with photosystem 2, even though it's counterintuitive, but photosystem 2 is breaking water down into hydrogen ions and oxygen ions. When it does that, it's extracting an electron. That electron is then used in photosystem 1. NAD is a carrier of an electron. So if you donate an electron to NAD, it'll become NADH. That NADH is a safe way to transport electrons to another location, which occurs in the mitochondria, which we're going to detail quite a bit. But that electron that's held in that NADH is eventually used in the mitochondria to produce ATP. ATP again, controlled charge separation. ATP in photosynthetic organisms is used to make plant sugars. So carbon dioxide is consumed by the plant, and those carbon structures are arranged to create sugar molecules, different rings. That's used for energy storage in plants, and it's used for structure, cellulose. Now when you get to the non-photosynthetic organism, non-photosynthetic organisms are still absorbing electromagnetic energy. This is going to be a very key point that I'm making in my biophotonic model. Conventional science only recognizes charge separation occurring within a photosynthetic organism. Non-photosynthetic organisms will take these plant sugars, and within the mitochondria of that cell, it's going to break those plant sugars down. In the process of breaking it down enzymatically, you will extract and make some ATP. So we'll detail it soon, but you're going to get only a little bit of ATP from breaking it down enzymatically. The key to this process is within the mitochondria, electrons are donated. So just like the plant created NADH within a mitochondria of a non-photosynthetic organism, NADH is produced in the mitochondria in the breakdown of sugar. That NADH is used to transport electrons. Now the important part to get to, which we're going to detail soon, is electromagnetic energy plays a key role in that transfer of electrons. So there's molecules, there's structures within the mitochondria that absorb electromagnetic energy. That energy absorption is used to make that charge separation for transfer. And that electron goes, we'll talk about it later, but it goes to oxygen, creating this. That oxygen negative is what's resulting in ATP production. So that it's just a chain reaction of moving that electron to a safer place. So what we're going to discuss next is how the mitochondria converts that NADH, that stored energy within those NADH, the electron that's donated, into ATP. So we're going to start off first by talking about glycolysis. So glycolysis can occur anywhere within any cell of the body. Any cell, even a single-celled organism, can ferment sugar, basically. What we're doing is taking glucose and we're enzymatically breaking it down into 2 pyruvates. So glucose is a 6-carbon structure. You're going to end up with 2 3-carbon structures. In the process of this breakdown, we need to donate 2 ATP to get the process started. What we're going to end up with is 4 ATP, and we're going to also end up with NADH. We're netting 2 ATP here. One glucose molecule has a capacity to make 34 ATP. Some models will say 36. We're going to stick with 34 ATP. So this very first part here, enzymatically creating those 2 ATPs, is not very efficient. It's not getting much of that energy that was stored in that glucose from, again, the photosynthetic reaction. So photosynthetic reaction stored that charge in here. We can extract that charge pretty easy in these 4 ATPs. After that, within the mitochondria, we go through another process called the Krebs cycle. The Krebs cycle is going to break these 3 carbon structures all the way down to carbon dioxide. In the process, no ATP is going to be produced, but we're going to be producing NADH and FADH. So these guys, again, as I said a minute ago, all they are is holding on to an electron. So in the Krebs cycle, basically we're taking these pyruvates, and as we're breaking them down, we're not releasing any ATP. There's not enough stored energy in there that's worth extracting. What they are doing is pulling off those electrons from the glucose molecule, actually from the pyruvate molecules, and they're storing that electron to bring to another location in the mitochondria to produce energy. So the next part is the important part. Within the mitochondria, we have 4 protein complexes and a final protein called ATP synthase. Now in the mitochondria, you have the outer membrane, but you also have an inner membrane. And then in the middle is the matrix of the mitochondria, the inside of the mitochondria. This is called the inner membrane space. So what's occurring here is these NADHs are going to come up to the first complex, and the FADHs are going to come up to the second complex. What they're going to do is donate those electrons to a structure called a cytochrome. We have multiple cytochromes within this process. So those electrons get donated to a cytochrome, and then those cytochromes are just going to transfer those electrons. When they transfer them, they're going to be pumping hydrogen ions outside into that inner membrane space. So what's happening is they're creating a positive charge and a negative charge. So these electrons are then going to transfer between the proteins. This is CoQ10. It's an acceptor and then a donator of an electron to another cytochrome. We're going to bounce. In between complex 2 and 3, there's a cytochrome called cytochrome C. Very important cytochrome. We're going to discuss it later, but that's the cytochrome that many models will say regulates how quick this process goes. It's the regulator. So these electrons continue to transfer through this process, pumping those protons out. This final cytochrome oxidase, has a very important job. It's going to donate that electron to oxygen. That donation is going to create water. At the very end, when that gradient has been produced enough, these protons are going to tunnel through ATP synthase. ATP synthase is actually a wheel. They say that tunneling of those protons through there spins the wheel. That spinning of the wheel is responsible for the conversion of ADP into ATP. Note, there's no enzyme there. They've never discovered this enzyme. They thought they've discovered it a few times, but nothing. So somehow there's just an energy transfer that creates this reaction. This is problem number 1 with our conventional model. This guy over here is going to be problem number 2. That's a lot of water. So they say 98% of the oxygen that we breathe is utilized in the mitochondria. So it's only making water. The other interesting note, they've never proved that this proton gradient actually exists. If it does, it's only minor. It's not a very strong gradient. So if you take a step back and you think about conventional model, we basically are storing an electron here. And the goal of the storage of this electron is to create another charge separation to create ATP. So we're going through this elaborate process of creating a very large charge separation in order to create 34 small charge separations. So one glucose molecule is going to yield 34 ATPs through this process. During this process, if things aren't going as planned, which they never do, every system has slight inefficiency. If during this process any of these electrons escape before they get to cytochrome oxidase to create water, we create something called reactive oxygen species. So basically you got an oxygen molecule that's holding on to that electron, and it's like a hot potato. It doesn't want to hold it. It's going to donate it to anything that will accept it. So things like DNA, proteins, and fats, if they interact with that, they're getting that electron. And that electron can change their structure and their function, even damage them. So we have a system within the matrix of the mitochondria of antioxidants. Antioxidants will accept that electron. So we have a buffer system. So this is known to be happening in conventional models. So our body anticipates it. It has a way to stop it from damaging other things. A side note, studies on aging. The aging process is the result of this. Since the system isn't perfect, you're going to have slight damage over time. The more you're utilizing energy, the more likely that you're going to have some type of interaction between reactive oxygen species and fats, proteins, DNA. So this system, very vital. Our dietary intake of antioxidants, not a huge part of this. I've read as low as 2% of our antioxidant capacity of our body is through dietary consumption. Not to say not to eat your antioxidants, but this is a very powerful system. Your body has a buffer to protect this. So this gets interesting because the reason I was studying this area is I was clinically having some experiences with red light therapy, laser, cold laser therapy. And I was utilizing it in ways that didn't quite make sense to me, but they were still working. We're going to detail my clinical experiences with it later. But I began to do a little bit of research on how is this possibly working? How is my cold laser stimulating nerve endings or acupuncture points, making changes that don't lie within this being a direct mechanism? And the reason I'm saying that is most of the studies on red light therapy and laser therapy, red light laser therapy, say the benefit is occurring because red light enhances the function of the mitochondria. So when you shine red light onto a cell, it's absorbing into these cytochromes. Cytochromes are actually light absorbing molecules. The center of a cytochrome is a heme molecule. Very complicated. It's a ring with an iron center. That heme molecule is the same type of heme molecule you'll find in hemoglobin. Oxygen will bind to that center, to that iron molecule, and it will hold there. So the theory of red light therapy enhancing this function is that when red light absorbs into these cytochromes, these guys are acting better, meaning they're accepting their electron like they should, holding onto it tight, and they're pushing it onto the next cytochrome. So there's less likelihood that we're going to get escape of electrons. More likelihood that we're going to efficiently pump protons out, and we're going to maximize our ATP production. There's one problem. Every study on red light therapy confirms it's increasing our ATP yield. It's speeding the process. It's allowing this to occur. But the other problem is it's also increasing reactive oxygen species. It makes no sense. If you're increasing the inefficiency of the system, how in the world are you producing more ATP? I sat on this topic. It was the very beginning of my research, and I sat on this topic for probably about 6 weeks. I was trying to figure out, there must be something else there. The reactive oxygen species levels correlating with increased output, there's something missing. There's a reason that this would occur. Couldn't find any. I ran into a lecture by a doctor out of India, an enzymologist. His name is Khalith, I'm probably going to say this wrong, Khalith Murali Manaj. Him and I discussed quite a few things after I had seen his video. He goes by Murali. He ran into a very similar situation where he was finding in his data, his research, that ROS, reactive oxygen species, weren't matching up with what should be happening. And he reran the data multiple times, kept coming up with this elevated ROS, also correlating with ATP. He came up with a new model. This new model explains the quagmire here. And the way it explains the quagmire is it's happening because they missed the first step of how the mitochondria functions. The mitochondria is not functioning by pumping these protons. This gradient, like I said earlier, it's never been proven to be that much of a charge separation that would be significant. This issue over here with the enzyme never being found. And we're just relying on the whole thing of transfer of energy. Well, what does that mean? You know, I'm experienced in the field of alternative medicine. I hear that all the time. Acupuncture works by chi flow, this energy. Well, what is this energy? It's this magical transfer that seems to occur even when ATP is consumed. And what I mean by that, when a cell is using ATP for energy, say in a muscle contraction, when that ATP cleaves, they're saying that this bond, this triphosphate bond, is a high energy bond. And when you split that phosphate off there, energy is released. What energy? Where's that energy even going? As a student, you kind of are learning this stuff and you're not thinking about it in stronger detail of what does this energy mean? How does ATP getting cleaved in a muscle contraction, what is that energy doing within that actin myosin filament bridge? It's changing electron flow, I guess, is the best they're going to get. You're donating that electron out. So the Merburn concept is saying that reactive oxygen species are actually the goal, that these cytochromes, make it bigger, these cytochromes within these protein complexes are taking those electrons from NADH and FADH and they're donating them to oxygen. They're creating this reactive oxygen species. Well, why in the world would they be doing that? Well, it just so happens that around the cytochromes, there's binding sites for ADP. So ADP is located right next to those cytochromes. So he described a process, it's called the Merburn process, where he believes that these guys are acting like enzymes in a way that we don't classically look at an enzyme. Classical enzymes have substrates which bind, and the enzyme facilitates the the catalytic conversion of those two different substrates into a new substrate. He's saying that there isn't any specific reaction going on there. You're just putting molecules, unbound ions, and free radicals in close proximity to each other and letting nature take its course. So what I mean by that, he's got a molecule, ADP sitting here. You got phosphate floating around within that matrix. And then you got reactive oxygen species. Those three things combined have an equilibrium reaction, meaning when they're all together, that oxygen that's sitting there, that electron sitting in that reactive oxygen species is going to interact with this, and it's going to result in ATP production. More importantly, he's saying that what happens within this system is you have what's called a MER zone. So around this protein complex, you got an area that's almost like a cloud of those reactive oxygen species. And those reactive oxygen species, if they're in the proper concentration, this reaction is going to move forward. If there's too much ROS or too little ROS, this reaction is not likely to occur. When that happens, now you have something called diffusible reactive oxygen species, meaning unreacted reactive oxygen species are now starting to seep into the cell. It's getting buffered by that antioxidant system I was just talking about, so those are neutralizing it, but if it's past its regular capacity, we're going to start having problems. The cell's going to start experiencing oxidative stress. Now when you shine a red light on that, why does that benefit it? I had contacted Morali about this, emailed him. I'm like, I'm working with red light therapy. I'm seeing that your model answers some questions I have. I understand how increased ROS is going to make ATP conversion happen. Where does the red light come in? Well, earlier I told you, these cytochromes with those heme molecules, they absorb electromagnetic energy. So that electromagnetic energy absorbing into those cytochromes is giving that cytochrome, exciting those electrons within that cytochrome to a level where donation of that electron occurs. So it just so happens that red frequency seems to do a good job of doing it. It's the optimal. And if you think about it in this perspective, if you put another frequency, let's say violet has a lot more electromagnetic energy than red does. So if you're starting to shine violet light on these cytochromes, you're activating those electrons to a much higher excited rate. You're going to create more ROS. Say if you shine near infrared light, and near infrared, so if you're looking at Roy G. Biv, near infrared is over here on the spectrum. As you go this direction, you're increasing that energy. There's more energy within that electromagnetic energy in the violet than there is the red. If you're shining near infrared into those cytochromes, you're actually going to slow that ROS production down. So that cloud's going to be at a lower concentration. So you're going to make less ATP, and you're more likely to have ROS escape because it's not getting consumed. I hope I made that concept clear and simple enough for you to understand that now we're looking at a situation where the conventional model on the most important part of cellular physiology obviously doesn't make any sense. It's not accounting for the charge separation that needs to occur with electromagnetic energy absorption within mitochondria. Mitochondria are dependent on electromagnetic energy. So in a single-celled organism, the DNA, your mitochondria, those single-celled organisms are going to absorb that electromagnetic energy. It's going to be a direct absorption. But now you start going into a multicellular organism, and you have layers and layers of cells. That electromagnetic energy of red light is not going to pierce very, very deep, maybe 1 to 4 millimeters. So how is that electromagnetic energy getting into the mitochondria deep within the cells? This is where things got very interesting to me, because the effects I was noticing clinically couldn't be described by benefiting just the surface of the body. I knew that electromagnetic energy was altering physiology in a whole different way. So when you get to multicellular organisms, you start coming to the conclusion that there must be a system within the body. There must be a way that electromagnetic energy is getting down into the musculoskeletal system. Because those guys are using a lot of energy. They're requiring a lot of ATP. If ATP production is dependent on electromagnetic energy to transfer that electron to oxygen to make reactive oxygen species, it's not going to cut it. It's only going to get this deep into the skin. So as an acupuncture, having acupuncture experience, traditional Chinese medicine experience, I started thinking, oh my goodness, is this what qi is? Is qi electromagnetic energy? Does the body have channels within the skin that transport this electromagnetic energy? That was pretty exciting to me, because for my entire career I was trying to join the concept of Chinese medicine, energy medicine, into an actual conventional model. You know, my practice is functional chiropractic neurology, so I'm working with basically stimulation, sensory stimulation of the brain. And I never put the two together that this system, electromagnetic system, could be tied to the nervous system until one day when I said I had some clinical experience, I was finding that using the cold laser on acupuncture points were affecting neurology. Well, that got pretty exciting to me, because then I started thinking, this system we're talking about of these meridians, these channels, this obscure thing that no one's ever proven existed, maybe it's less obscure. Maybe this system has to do with nerves. So sensory nerves in the skin, is it possible that those are absorbing this electromagnetic energy? And that our nervous system not only has a binary component of nerve firing, nerve not firing, maybe this system works with electromagnetic energy. That's when you start getting into the quantum biology aspect, that maybe there's a quantum nature to the body. When you think about a multicellular organism, I kind of like to think of the nervous system, your brain, your spinal cord, your nerves, having communication with all the cells of your body. Well, how does every cell of your body cooperate within a role to have one function as one organism when there's no possible way these nerves contact every cell? The nerves are going to be between cells, and there's going to be multiple layers of cells on each side of the nerve. There is not direct contact of every cell to our brain. So when I started getting into this thought process, maybe electromagnetic energy is traveling through these nerves. Maybe these nerves emit electromagnetic energy. Maybe this system that I discovered has more to do with communication than it does with just energy production in the mitochondria of the muscles. So I started experimenting, where I was using the laser on acupuncture points. I stopped using them on acupuncture points. I started using them on nerve dermatomes. So I would do a stimulus on the nerve dermatome. I'm sorry, I'm going to say this a different way. I would do a stimulus on an acupuncture point where I knew it had a neurologic effect. I would also do that same stimulus a little bit over from the acupuncture point on either direction, and it still functioned, meaning there was no significance of having that laser specifically on an acupuncture point. That effect happened anywhere that I stimulated that particular sensory nerve. So I started to play around with this, and I was using the laser on different, we call them dermatomes. So on the skin, you're going to have a certain representation for each spinal level. So the C2 spinal nerve is going to have a certain representation on the skin. You're going to have a C5 representation on your skin. I started looking at these acupuncture meridians, and I started finding that at the distal points in our hands and in our feet, those meridians were matching up to dermatones, meaning what the Chinese were thinking were acupuncture channels were probably just those dermatones, and that qi, what they thought was qi, was actual energy flowing through that system, the nervous system, I should say, and not some mystical channel that's under the skin that nobody knows what it actually is. So when I made that connection, that what the Chinese thought of as qi is probably electromagnetic energy, and it also represents on the nervous system, that was a pretty important breakthrough in the concept. That's when I started creating what my book is about, that we have a biophotonic system within our body. That biophotonic system is necessary because I know that electromagnetic energy has to get to these mitochondria. But I think it's much more complex than that. There is a lot more interactions of electromagnetic energy with other aspects of the cell. Energy production was just the beginning. So we blew through quite a bit of information on the very first one, and I pretty much covered a lot of stuff in a scattered direction. The next videos, I'm going to break down specific areas and how this system coordinates everything, how charge separation through electromagnetic energy produces a system that keeps the body in coherence, that allows cellular communication between the whole organism, how it allows energy production within the organism. This electromagnetic energy in the Merburn concept goes way beyond just the mitochondria. That Merzyme reactive oxygen species concept can be applied to liver detoxification. It can be applied to hormone production. Dr. Morali, I haven't read all his papers, but he lists tons of areas where he believes that this diffuse reactive oxygen species, Merzyme concept, explains a lot of ways that different functions occur in the body that don't fit the classic enzyme role, which I'm going to quick talk about. So I'm going to go through the difference between what a Merzyme is. So this would be, we're going to say, one of the protein complexes within the mitochondria, creating that cloud of reactive oxygen species to allow the interaction of ADP, P, and reactive oxygen species. So this is called a non-probabilistic reaction. You're just basically putting together everything that needs to be in place and letting the reaction occur. If you think about it, this is how things started. Life was not very complex in the beginning. Non-probabilistic reactions are what were creating the early processes of life. As things started getting more routine, where there was reactions that were repetitive, things that the organism knew it would have to perform, we started coming up with classic enzymes. A classic enzyme is going to bind the two substrates I was talking about before, and it's going to cause an interaction. So A and B are going to join, and you're going to have C. Deterministic, meaning this particular enzyme is going to have this particular reaction every time it interacts. How this happens is going to get pretty interesting, because I think electromagnetic energy is going to play a role in this, which will be more detailed later. But the reaction that's happening here is still going to be an electron transfer. It's still going to be taking charge separation and manipulating one of those substrates so it does interact with the other. Any enzyme is either taking electrons away, putting electrons on. When they're doing that, it creates the ability for two molecules that would not have interacted before, because say these molecules were both, these are both charge neutral molecules. So if they're bumping into each other, the likelihood that those molecules are going to interact and combine to each other is just not going to happen. But if you make one of these guys negative or positive, well, now something's going to happen there. That's going to create the catalytic reaction that we're trying to get. So, very important concept that's going to be talked about quite a bit. This is I'm sorry, this is deterministic. Deterministic response, non. It's going to play a part in the communication system, the biophotonic system I'm talking about too. What is our nervous system? When I say that the nerves are a binary nervous system, those nerves are either firing or they're not firing. So there's a reaction that's going to be, how should I say, deterministic. So you're going to get, if a nerve fires to a muscle, you're going to get a contraction of a muscle. So the nervous system also could be thought of in a deterministic way. The quantum system, the system that I'm going to discuss in detail soon. When I was saying that the electromagnetic energy is flowing through the system, through the nervous system, it's going to have a non-probabilistic reaction, meaning depending on the frequencies of energy, what combinations there are, that's going to determine how this quantum system reacts. It's not going to be so cut and dry as this. So the nervous system, when it's working, is having a specific outcome. When the quantum communication system, the biophotonic system that I'm discussing, that the whole topic of my book, this quantum system has a lot more probability of reactions, meaning multiple outcomes can occur. So it's more adaptable than this basic nervous system. Just like an enzyme, it's not very adaptable. So if we expose ourselves to a new toxin of some sort, they decide to put whatever in your food tomorrow that's new, and you go to eat it, your liver is not making these enzymes, the enzymes to break down that new toxin. But within those liver cells, there's still this non-probabilistic reaction that can metabolize that toxin. It's not specific to a toxin. And what's it doing to that toxin? The same thing this is doing to the toxin. This is going to interact with a known toxin, bind it, add an electron, add something to it to change its structure, make it water soluble, not harmful anymore, and you're going to get rid of it from your system through your urinary tract. Over here, doesn't matter. You're dealing with these reactive oxygen species. So if you're binding particular types of molecules, and that's what a liver merzyme would do, it would have an affinity to certain types of molecules that it knows are just problematic molecules. It will hold it within this Dross zone, and it will neutralize it. It will add that electron, it will add another charged particle, an ion, and neutralize it. And it usually is a phosphate or a glycine. It's usually a phosphate. My detox background isn't so good, but people out there more familiar will understand what I'm saying. You're basically just adding some type of particle to that toxin to neutralize it. So that's going to complete what I wanted to discuss in this first video. I'm always open for questions. The reason I'm producing these videos is for my patients who are interested in trying to understand my book, and then for people in the field that are trying to figure out this system. I'm always open. Email, I'll have it on the links. If you want to know more, obviously my book is going to be in much more detail. Purchase a copy. They're on Amazon. I'll also create a link for that. And yeah, we're going to keep walking through this series. Some of these topics are going to go more detailed. Some of them are going to be pretty simplistic. But just keep in mind from what we talked about today, charge separation. Very important to life. Everything in life is about charge separation. And that first charge separation is coming from harnessing electromagnetic energy within pigment systems like the cytochromes, the chloroplasts. So once that charge separation is harnessed, that's when things start going. Thank you. I'll talk to you soon on our next video.